Welcome back, class. This is Silvana O'Coney with Lecture 8, Antimicrobial Chemotherapy. Now, we know that the control of microorganisms is critical for preventing disease and treating disease. But antimicrobial compounds are also incredibly important tools used in research and also in industry. Now, we've looked at the use of chemical and physical agents, and when disinfecting or sterilising an inanimate object, one naturally has to use procedures that don't damage the object itself. The same is true for treating living organisms. The most successful antimicrobial compounds will interfere with vital processes that differ between the microorganism and the host. Thereby, it seriously damages the target microorganism but harms the host as little as possible. So this lecture is going to introduce the principles of antimicrobial chemotherapy, and briefly we'll look at the characteristics of selected antibacterial, antifungal and antiviral compounds. One of the most serious threats to public health in the 21st century is the spread of drug-resistant pathogens. Bacteria often become resistant in several different ways, and once resistance originates in a population, it can very easily be transmitted to other bacteria, even different species of bacteria. Unfortunately, a particular type of resistance mechanism is not confined to a single class of drugs. So two bacteria may use different resistance mechanisms to withstand the same chemotherapeutic agent. Furthermore, resistant mutants arise spontaneously and are then selected for in the presence of the drug. The long-awaited superbug arrived in the summer of 2002. Staphylococcus aureus, a common but sometimes deadly bacterium, had acquired a new antibiotic resistance gene. The new strain was isolated from foot ulcers on a diabetic patient in Detroit, Michigan. Methicillin-resistant Staphylococcus aureus, MRSA, had been well known as the bane of hospitals. But this newer strain had developed resistance to vancomycin, one of the few remaining antibiotics that was still able to control Staphylococcus aureus. This new vancomycin-resistant Staph aureus, otherwise known as VRSA, also resisted most other antibiotics, including ciprofloxacin, methicillin, and penicillin. Isolated from the same patient was another dread of hospitals, vancomycin-resistant enterococci, VRE. And they did some genetic analyses which revealed that the patient's own VRSA had acquired the vancomycin resistance gene from the VRE through a type of bacterial mating called conjugation. So was born a new threat to the health of the human race. Now let's take a look at the different mechanisms used by microorganisms when they develop resistance. Let's start with the first one listed here. So many microorganisms often become resistant simply by preventing entrance of the drug. For example, many of the gram-negative bacteria are unaffected by penicillin G because the molecule simply can't penetrate the envelope's outer membrane. Genetic mutations occur that lead to changes also in penicillin binding proteins, and this can render a microbial cell resistant. A decrease in permeability can lead to resistance to the sulfonamides. For example, mycobacteria resists many drugs because of the high content of mycolic acids. And these acids are found in a complex lipid layer outside the peptidoglycan. This layer is impermeable to most water-soluble drugs. Other mechanisms listed here include pumping the drug out, inactivation of the drug, altering the target enzyme or organelle, which is normally affected by the drug, and use of alternative pathways or increased production of target metabolite. Let's have a look at these a little bit more closely on the next slide. Here we have a schematic of a bacterium which illustrates some of these mechanisms of resistance. Let's start with number 4. So go to number 4 on this image, which is towards the upper part of the cell. And this illustrates a second resistance strategy, which is simply to pump the drug out of the cell after it's actually entered. Some bacteria have plasma membrane pumps that actually expel drugs, and because they're relatively non-specific, they can pump out many different drugs, and so these transport proteins are often called multi-drug resistant pumps. Many of them are actually drug proton antiporters, that is, protons enter the cell as the drug leaves. Now let's look at mechanisms number 2 and number 3, which are very closely related. And these two mechanisms illustrate the way in which microorganisms resist attack by drugs through chemical modification. The best known example is hydrolysis of the beta-lactam ring of penicillins by the enzyme penicillinase, which we've discussed earlier. Drugs also can be inactivated by the addition of chemical groups. So, for example, chloramphenicol contains 2 hydroxyl groups, and these can have acetyl groups added to them to inactivate them. Another example involves the aminoglycosides, which can be modified and thereby inactivated in several ways. So, for example, there are enzymes that can catalyse the addition to the hydroxyl groups of either phosphates or other chemical groups to inactivate them. So we can see in number 2 the approaching antibiotic in blue, and in brown we have molecules which are antibiotic degrading enzymes. So these enzymes have been secreted from the bacterial cell and they degrade the antibiotic before it even enters. Mechanism number 3, which is within the bacterial cell, you can see the antibiotic has actually entered the cell, that is the little blue triangles, and they're immediately wrapped up and modified by these molecules in green. And these are antibiotic altering enzymes which would do the chemical modification. Now we can look at mechanism number 1, so towards the bottom part of the bacterial cell. And in this example, the antibiotic represented by the blue squares are entering the cell, and they would normally target the cell ribosome. But resistance arises here when this target cellular structure is modified so it's no longer bound by the actual drug. Finally, a final mechanism, resistant bacteria may either use an alternate pathway to bypass the sequence that's inhibited by the agent, or it might increase the production of the target metabolite that's being affected. So, for example, some bacteria are resistant to sulfonamides simply because they use preformed folic acid from their surroundings rather than synthesizing it themselves. Other resistant strains arise because they simply increase the rate of folic acid production and thereby counteracting the inhibition by the sulfonamides. So let's consider the question, how do these resistance mechanisms arise and how are they transmitted from one microorganism to another? To begin to understand this, we need to look at resistance genes. Resistance genes can be found on bacterial chromosomes or on bacterial plasmids or within transposons. And when they're found on mobile genetic elements, such as plasmids and transposons, they can be freely exchanged between bacteria. Occasionally, mutations in the bacterial chromosome occur, and these can make the bacteria drug resistant. Usually these mutations will result in a change in the drug target. Therefore, the antibiotic can no longer bind and inhibit growth. For example, the protein target to which streptomycin binds on bacterial ribosomes. Many of the mutants that arise are probably destroyed by the natural host resistance mechanisms. However, when someone's been treated extensively with antibiotics, some of the resistant mutants may survive because they now have this competitive advantage over the non-resistant strains. Often a bacterium is drug resistant because it has a plasmid. It contains a plasmid, and that plasmid contains one or more resistance genes. These plasmids are called R plasmids, as you can see here, where the R stands for resistance. These plasmid resistance genes often code for enzymes that destroy or modify drugs. So, for example, the hydrolysis of penicillin or the addition of acetyl groups to chloramphenicol and so on. So once a bacterial cell possesses an R plasmid, the plasmid or its genes may be transferred to other bacterial cells quite rapidly through normal gene exchange processes such as conjugation, transduction, and transformation. And because a single plasmid might carry genes for resistance to several different drugs, a pathogen population can become resistant to several antibiotics at the same time, even if a person's only being treated with one particular drug. Resistance genes can also be located on mobile genetic elements other than plasmids. These genetic elements are called transposons, and they contain also genes for antibiotic resistance, and some of them actually contain more than one resistance gene. These are found in both gram-negative and gram-positive bacteria, and they can move very rapidly between plasmids and through a bacterial population. So the extensive use of antibiotics actually favours the development of resistance because the drug destroys the susceptible bacteria that normally outcompete the drug-resistant strain. Several strategies have been proposed to reduce the emergence of resistance, and they are listed here. Firstly, the drug is given in high enough concentrations to destroy susceptible bacteria as well as any mutants that may arise spontaneously during the treatment. Second strategy involves the use of 2 or 3 different drugs at the same time. And this is done with the hope that each of the different drugs will prevent the emergence of resistance to the other. The third strategy here is that these drugs should only be used when absolutely necessary, and if possible, the pathogen should be identified, drug sensitivity tests run, and the proper narrow spectrum drug should be used. Despite these efforts to control the emergence of resistance, the situation continues to worsen. So pharmaceutical companies continually collect and analyse samples from around the world to search for new antimicrobial agents. And a very interesting approach is based on an idea that was first proposed early in the 20th century by Felix Stirrell. And you might remember that he was one of the discoverers of bacterial viruses, otherwise known as bacteriophages. He proposed that bacteriophages could be used to treat bacterial diseases. But most microbiologists didn't actually pursue this idea, probably because of the introduction and discovery of antibiotics. However, Russian scientists did develop the medical use of bacteriophages. Currently, they use bacteriophages to treat many different bacterial infections. Bandages are saturated with phage solutions, phage mixtures are administered orally, and phage preparations are given intravenously to treat bacterial infections. There are currently 3 American companies that are actively conducting research on phage therapy. Let's turn our attention now to antifungal drugs. So treatment of fungal infections generally is less successful than that of bacterial infections, and this is mostly because eukaryotic fungal cells are much more similar to animal cells than are bacteria. So many of the drugs that inhibit or kill fungi are also quite toxic for animals. In addition, most fungi have a detoxification system that modifies many of the antifungal agents. So as a result, the added antibiotics are fungistatic only as long as they're applied repeatedly. Effective antifungal agents frequently either extract membrane sterols, thereby disrupting the membrane, or prevent their synthesis. Fungal infections are often subdivided into infections of superficial tissues, known as superficial mycoses, as written here, or systemic mycoses. And treatment for these 2 types of mycoses is very different. So systemic infections are much more difficult to control than the superficial mycoses. Okay, shown here are 2 of the 3 drugs that are commonly used against systemic mycoses. To the left we have amphotericin B, which will bind sterols in the fungal membranes. And to the right we have flucytosine, which acts by disrupting RNA function. These are actually quite toxic, both quite toxic substances, and normally used only for serious infections. Let's finish now by looking briefly at antiviral drugs. So drug development has been slow in this area, and that's because for many years the possibility of treating viral infections with drugs appeared remote, because viruses enter host cells and they make use of host cell enzymes and other components. So it was thought that a drug that would block virus reproduction would also be toxic for the host. But now inhibitors of virus-specific enzymes and virus life cycle processes have been discovered, and several drugs are used therapeutically. A couple of examples are shown here.